Thank you,
Thank,
Dr. Teresa. Thank
thank you once again.
you. Moving to our next talk. The next talk is by Dr. A. Usha Raja Nanthini, a distinguished professor and head of the Department of Biotechnology at Mother Teresa Women's University, Kodaikanal. Uh, she's been 19 years of academic excellence and leadership with a doctorate in philosophy in biotechnology from Periyar University, Salem, and an educational journey that spans across several prestigious institutions. Dr. Usha stands as a beacon of knowledge and innovation in her field. Her academic credentials are further bolstered by a Master of Philosophy and Master of Science in Biotechnology alongside a Bachelor of Science in Zoology, showcasing a broad and deep understanding of biological sciences. Dr. Usha's scholarly work is recognized globally with contributions to various national and international seminars and conferences, totaling 52 papers. Her leadership extends beyond academia, evident in her roles as an executive council member, control of examinations, director of distance education, and several other administrative capacities within her university. Dr. Usha's commitment to education research is further demonstrated by her involvement in numerous committees and councils aiming to elevate the standard of education and research in biotechnology. A visionary in her domain, Dr. Usha has initiated multiple memorandums of understandings with international and national organizations, fostering research and teaching collaborations that bridge gaps and build networks. Her dedication to the advancement of biotechnology and education reflects in every aspect of her career, making her an inspirational figure for students and peers alike. I welcome Dr. Usha for her talk on fungal cosmeceuticals. Dr. Usha? Yeah,
Thank you, Dr. Rohit Sharma, sir,
yeah.
for nice introduction. Can you hear me, sir?
Yes, yes, you are audible, ma'am. Clearly audible.
Yes, sir. So before I start my session, I would like to thank Dr. Baladamudashana, sir, from MycoAsia, MycoIndia, for providing me this opportunity in the midst of legends of researchers in mycology field. And really, I'm glad to be here to present this small session on the fungal cosmeceuticals.
Would you like to share your slides?
Yes, sir. Is it visible, sir?
Uh,
Madam, you can minimize the browser. I think you have to share again.
Is it visible, sir?
Madam, you minimize the browser. Minimize the Chrome browser. Oh yeah, now it is visible, madam.
Yes, sir.
yes.
Madam,
Yeah.
can you just hide that notification? That is okay. Thank you so much. Yeah, please.
Yes. Yes. So actually, I'm from Department of Biotechnology, Mother Teresa Women's University. So this Kodaikanal actually uh, is in Dindikal district, which is there in the Western Ghats. It is one among the biodiversity hotspots, and really, it is actually in the midst of treasure of resources. And this is our campus, university campus. It is actually in a very rural based area, which is having many rural hamlets around and many rural and tribal hamlets around this campus. And it is totally away from cities, and this institution is only for women, the research, teaching, and extension for only women. And here, actually, we started our research in versatile aspects. We are indulged in activities like anti-corrosion studies with myco-synthesized nanoparticles. We have some hybrid strains made by interspecific protoplast fusion between filamentous fungi and mushrooms. And actually, we are not taxonomists and biotechnology department. We are involved in application-oriented research. So you can see some patents published recently, like wound healing ointment from macrolepiota extract and a plant-based refreshener releasing perfume along with anti-dermatophytic effect and wall protective effect. So like this, we are indulged in versatile application-oriented works with fungi and mushrooms. And in 2019, we were blessed with a fund released from DST for exploring more about the potentials of wild mushrooms of Kodaikanal. So when navigating through that, through various researchers with the funded support from the DST, I was fascinated, actually got fascinated with this topic, fungal cosmeceuticals, because today economy is knowledge based. So I am also in charge of IPR cell of our university. So I know the importance of IPR and the knowledge based economy, how much is important to India, and when we are in the era of demographic dividend. So really, I was fascinated towards this topic, fungal cosmeceuticals, and I want to create some awareness to these young researchers of our country. To at least some of them hearing this topic will take up some research and end up with some very wonderful products with the help of fungi. So let me now enter into the topic. So I'll give some glimpse what is the necessity of this topic today, and then proceed with. So actually, it is not new. This cosmeceutical topic is not new. So archaeologists discovered sharpened red ochre sticks for coloring the body at one Homo erectus site. So Homo erectus, they lived 1 million years ago. And same way, this Neanderthal sites. So those Neanderthals who lived 30,000 to 1,25,000 years ago, they painted their dead with red ochre before burial. So this all archaeologists have discovered, and it shows that cosmetics has played major role even in those days. So now, when we think about Cleopatra or Pharaohs, when we can imagine that malachite yellow shade what they used, the makeup they used. So till today, we can imagine by some literatures, the photographs, what we see, paintings we see, we imagine their makeup and eyeshadows. So the ancient Egyptians, Greek, Romans, so in their culture, they have used this personal care products long back. So actually, as cosmetology, as ancient science, so it has come out from Egypt and India. So early records of cosmetic substances and their applications is there in Indus Valley civilization long back from 2500 and 1500 BC. So the early records are from Indus Valley civilization in India. So it is this record shows that they had highly advanced idea of self beautification and cosmetic usage for both men and women. This cosmetic usage practice was interwoven with seasons and the normal rituals of life. And when we go to the Egypt side, the women have used this cosmetic products 5,000 years back. The Greeks and Romans, they applied massage oils on large scale, and they had written book on preparation of cosmetics through the introduction of distillation and extraction processes. So later on, then gradually Europeans picked up and it came to know to them. And until 1900s, it did not come to ordinary citizens. Only royal families were using pigments, colors, cosmetics, and flavoring agents, and so on. But today, cosmetics and personal care products a sign of civilized society. And so I can say with examples how cosmetic has become important and how it is going to rule the future. The article Middle Class in India from 1947 to the present and beyond, published by the Association for Asian Studies in 2018, says that the middle class population in India was about 5 to 10 percentage of total country's population in 2005, and it is expected to increase about 90 percentage in 2039. So when this middle class population percentage increases, they prefer high quality and premium cosmetics. And additionally, this rising income will propel this demand. And next, again, the statistics say that the demand for cosmetics is increasing because there is number of working women increase in the whole world. So, example, a data from Australian government 2020, among all employed individuals in Australia, 47% is women. So, it's not only in Australia, worldwide the scenario is same, and India is no more exceptional. And again, see, Indian Express says Miss World 2019 had estimated to have a viewership of 900 million from 140 countries. So it shows that the beauty contests like Miss World, Miss Universe, and Victoria's Secret generate attraction for cosmetic products. And not only this, there is again this another reason for the demand. Survey on world population aging 2019 reported by Department of Economic and Social Affairs of United Nations in 2019 says nearly 164.49 million people in China were in the age group of 65 years and above. So not only in China, Japan and few other developed countries, they are having very large pool of aging population. And because of this very large pool of aging population, there is demand for anti-aging, anti-wrinkle, and dark spot treatment products. So data from Ministry of Economy, Trade and Industry, Japan 2019 says the production of cosmetic product has raised from 434,000 tons in 2017 to 455,000 tons in 2018. And again, another reason. So see the pictures which I'm displaying here. So this dark skin girl, so we will agree, even though she's having a dark skin, she's very beautiful, and even these pics too. And Miss Angola, Laila Lopes, she was crowned Miss Universe in 2011. So world agrees even dark skin are beautiful, but worldwide efforts have been made to find more effective skin whitening agents, especially for Asian countries. And they think white is more beautiful, and they are attracted towards this white skin whitening agents. And brown spot and irregular patches are considered abnormalities, and there is demand for this. This makes Asia Pacific dominate global cosmetic market, and it is valuated that 112.57 US dollar billion, 112.57 billion US dollars market in 2022. So Asia Pacific dominates the global cosmetic market. And in global level, in 2022, the market of cosmetics was evaluated as 299.77 billion US dollars, and it is projected to grow 417.24 billion US dollars by 2030. So this all surveys shows the same thing. They say this much hike is going to be there by 2030. And again, rising preference towards organic formulations has accelerated this research and development activities. And why I'm here to take this topic here is also based on this demand for organic formulations. US Organic Industry Survey 2020 made by Organic Trade Association says total sale of organic non-food has raised 9.2% just in 1 year from 2018 to 2019. In 2019, it was 5 billion US dollars, and it has rise 9.2 percentage within 1 year. So throughout the world, researchers and cosmetic industrialists now continuously seeking for new, so this again I should highlight, new, safe, better natural compounds with higher potentials when compared with the artificial synthetic ones available today in the market. There is increasing trend for sun care products, night skin repair creams, refreshing fragrances, hair colors, and others. So when you see the share of the cosmetics in global market, the highest the share is there for this skin care. So now before entering into the topic, I'll just give a glimpse about what is cosmeceuticals. Actually, what is cosmetics? A substance or a product that can be gently applied on various parts of the body, either by rubbing or sprinkling for cleaning, conditioning, protecting, beautifying, enhancing attractiveness. And moreover, it should not exert any side effect. So these things, these products or a substance will come under the general term cosmetics. Again, this cosmetics, it is in two types: cosmeceuticals and nutracosmetics. So what are cosmeceuticals? This, the product or the substance with all these effects, with all these characteristics, but which can be applied topically, like creams, lotion, and ointments. They fall under this category, cosmeceuticals, which for cosmetic purpose, if it is ingested orally to give shine to the skin or beautifying this overall appearance, when it is ingested orally, it is known as nutricosmetics. But now today we are only dealing with cosmeceuticals, and most important one in this is skin whitening. Actually, the synthesis of pigment melanin determines the human skin color. That is responsible when there are people in the country with more sunshine, they have more melanin in their skin and their skin is dark. So this is the overall thing behind actually, and how these creams are prepared and where is the target. I'll explain you now. The factor induces the melanocytes to oversynthesis melanin, continuous exposure to UV radiation, autoimmune disorders, genetic factors, change in hormones that stimulate melanocytes, and some few therapeutics and medicines with side effect and radiations. This all make the melanocytes to oversynthesize melanins. So this is the pathway. See here, tyrosine is the converter to dihydroxyphenylamine, that is L-DOPA, with the help of tyrosinase enzyme. This tyrosinase actually will get converted into eumelanin, that is brown black pigment or yellow pigment with some other enzymes. And this pathway, whether it should go producing yellow pigment or brown black pigment, is decided by the physiological condition. Here, the tyrosinase enzyme is rate limiting step, which will decide. So this very important enzyme, which is inducing melanosynthesis, or it regulates actually the whole pathway. So reduction or prevention of melanogenesis can lighten the skin color. So this is the basic principle behind. So this is the pathway. So reduction and prevention of this melanogenesis can lighten the skin color. So when this is the theme, the enzyme tyrosinase, which is contributing for this rate limiting step, regulates the biosynthesis pathway for melanin formation. So the target now for this products, skin whitening products, is on this tyrosinase enzyme. So this tyrosinase inhibition is the target. So when tyrosinase is inhibited, the melanogenesis is inhibited and the skin color can be lightened. So usually we have this cream for this purpose, hydroquinone, but it is reported that it is having carcinogenicity. It exhibits exogenous ochronosis, cell irritant property it has, and unpleasant fish odor. So now the researchers, the industrialists are searching for alternative product. Again, we have kojic acid and corticosteroids for the same purpose, but it is also reported when long-term usage is done with these products, it is having some adverse effects. Now we will come to our fungi as skin whitening agents. Actually, in 1895, the first report from Rusula nigrinens, when it was cut and exposed to air, it turned red and then to black. And the reason when found, it was because of the tyrannase enzyme. And later on, studies found that all phylogeny from bacteria to mammals have this tyrannase enzyme. And this tyrannase enzyme in the fungi catalyze the first rate limiting and centralized step in melanogenesis, is conversion to black, is by this tyrosine to melanin. Again, when studies proceeded, they found Agaricus bisporus and human tyrosine enzyme showed highest similarities, and tyrosinase as model for melanogenesis studies. So to conduct tyrosinase, this melanogenesis studies, tyrosinase obtained from Agaricus bisporus will be helpful, they found out, and mushroom tyrosinase became available commercially. Now, all the studies, we know that most of the organic, non-chemical cosmetics coming out are plant-based, and when they conduct experimental studies to related to inhibition of tyrosinase, they use commercial mushroom tyrosinase. So it is available commercially. And so for all the studies conducted on this tyrosinase enzyme is obtained from mushroom. Sorry. So far, the studies conducted on fungi anti-tyrosinase effect has come from these mushrooms. So from these mushrooms, reports are available with anti-tyrosinase effect. Ganaraballicidum, lentinus edodes, pleurotus, nabradensis, agaricus brasilensis. So this all mushrooms, the reports are available with anti-tyrosinase effect. And even from this fungi, reports are available, and commercial compounds like gallic acid, hispolan, caffeic acid, and nicotinell, tyramine, and paracaumaric acid, and this all compounds commercially available which are obtained from fungi. The reports are available with anti-tyrosinase effect. So now fungi has potential with anti-tyrosinase effect, and which can serve as very effective skin whitening agents. And apart from that, there are some reports like bioactive compounds isolated from Agaricus bisporus, Inonotus obliquus, Metarrhizum anisophalae, and exopolysaccharide from Gryphola frondosa. So this also has shown anti-tyrosinase effect. And Aspergillus niger, actually it is reported to ferment plant tannins to produce polyphenols like ellagic acid and gallic acid. These are also effectively used as depigmentation agents. And lactic acid produced from rhizopus species also been used as skin whiteners. Again, one more thing. So this sake production. So when sake is produced from rice, they make koji. So with yeast as starters, koji is prepared. This kojic acid is reported with the whitening agent, and so far it was used. And earlier I said it, when it is used longer time, it has some adverse effect. And this tyrosinase inhibitory activity was many-fold higher than the kojic acid in protocatacoldehyde obtained from the mushroom, this one, Philinus lintus. So this report is also available. So this shows more potential is there with this fungi towards this skin whitening agent. And Sisophyllon, again, from Sisophyllon camine, is reported to minimize skin damage caused by UV and exposure to skin to toxic and polluted environment. Next important aspect as cosmetic is anti-aging potential. So fungi also has anti-aging potential. So before that, we'll see how it works and how fungi can contribute to that. So skin is the largest organ in human body, which gives a barrier against invasions. The skin has layers epidermis, dermis, and hypodermis. This epidermis layer is actually having the melanocytes, and it is attached to dermis layer beneath it by connective. The structural and mechanical support to the skin is given by extracellular matrix. So this skin extracellular matrix gives structural and mechanical support to the skin, which protects the integrity of the skin. So this we have to remember. And this extracellular matrix has two structural proteins. The extracellular matrix has two structural proteins, collagen and elastin, produced by the fibroblasts, the connective tissues present in this skin produces this collagen and elastin. And they are very important for skin aging process. So when the skin ages, it gets wrinkles, age spots, it becomes rough, elasticity lacks, saggy, and laxity skin. This is the process of skin aging. So this is because of genetic factors. This is intrinsic skin aging is by these genetic factors, and external factors make skin aging. It's because of overexposure to UV radiation and toxic environment. They cause oxidative stress, and that leads to extrinsic aging. And reduction in collagen levels. It may be either due to increased degradation or declined synthesis. This reduction in collagen also may lead to the aging process. Again, the zinc-dependent matrix metalloproteinases, which are involved in extracellular matrix degradation, increase the secretions. Matrix metalloproteinases secretions will be increased, and this also is noted with increasing age. The fibroblast number, which is the connective tissues present in the skin, and their capacity to synthesize procollagen are also found to reduce aged skin when compared with young skin. So these are all the factors involved in aging process. So extrinsic aging factors, intrinsic aging factors, and they involve in reducing or degrading the collagen, which will lead to aging process. So you can see here the young skin, how it is, the elastic fibers, collagen, how it is there in the young skin, and it is broken and it undergoes atrophy in aged skin. So hyaluronic acid, elastin, collagen. So these three in the extracellular matrix support cells to regenerate and repair itself. Here comes these enzymes, elastase, collagenase, hyaluronase enzyme, and other matrix metalloproteinase enzymes involved in this degradation process. Here, in cosmetics, these are the targets. So when these play in degradation or in decreasing the levels of elastic fibers, elastin, and hyaluronidase, collagen, so these enzymes are responsible for that. So cosmetic industries and researchers, they target these enzymes. Inhibitors of elastase, collagenase, hyaluronidase, and other matrix metalloproteinase enzymes have become the target for skin aging treatment. So now I'll show you this fungi, mushrooms, the extracts of these mushrooms have been reported to have anti-hyaluronidase effect. So it is very, very important. Hyaluronidase property is very important to skin and other tissues because it is having very major role in skin rejuvenation, moisturization, viscosity enhancement, and permeability. So when this is destroyed, so the skin aging, it lead to skin aging. But creams and other lotions with anti-hyaluronidase property will reduce the skin aging effect. So this extracts from mushrooms have been reported already, and similarly anti-elastase and anti-collagenase property. So these two are also very important for skin, young skin, and these mushrooms, the mycelial extracts and exopolysaccharides isolated from these mushrooms have anti-elastase and anti-collagenase property, and phenolic compounds and polysaccharides isolated from Olvaceae has anti-collagenase effect, and ergothioneine found in certain fungi is a derivative of histidine amino acid. It inhibits expression of MMP1, and this most ergothioneine is obtained from Portabellas, Crimnus mushrooms, and Agaricus bisporus. These reports are available, and some hybrids are produced with combination of paracoumarate, ergothioneine, ascorbic acid. This hybrid prepared with coumaric acid, they also has reported to increase the formation of collagen in human dermal fibroblast. Next comes this fatty acids, the outermost layer of skin that protects against oxidative damage is a complex structure with components like fatty acids. So intracellular matrix gets supplemented by fatty acids. It inhibits the degradation of collagen and improves cell function. So fatty acid is next target actually. It is very easy because fungi have very small lifespan and it can be produced in a large scale very easily. So fatty acids can be produced by fungi and used in cosmetics as antioxidant, cell regulators, anti-inflammatory agent, and to promote healthy skin growth. So this fungi, Pythium and Mortadella, have been reported for producing omega-3 fatty acids which regulate inflammatory response, which prevent skin rashes and redness. And omega-6 from these fungi and mushrooms, omega-9 again, it is found in cell membrane, abundant in skin, and it serves as excellent medium through which nutrients get transferred, and it's very, very, very important for skin rejuvenation, and it is obtained from these fungi. And extracts of this tubers have been reported as effective regenerative, very good potential regenerative formulations from these tubers. And next comes as a cosmetic, is a moisturizing agent. Fungi again has very good potential for this moisturizing effect. The skin need this moisturizing effect because appearance and smooth functioning of skin depends on the surface lipids and water content present in the stratum corneum. So moisturizers are those increase the skin humidity and enhances the surface physicochemical properties and keep the skin moist, smooth, and less prone to wrinkle formations. So actually traditional moisturizing agents use pantothenic acid, ascorbic acid, and hyaluronic acid as common ingredients. And reports shows with the help of fungi and mushrooms, the extracts from mushrooms and fungi has shown that it retains moisture than that which is used in traditional moisturizing agents. That is hyaluronic acid or sodium hyaluronate, which is used traditionally as a common ingredient. Than that, the reports have shown that the fungi and mushroom extracts are very effective and positive than traditional moisturizing agents. And lactic acid production from rhizopus strains, they are also helpful as ingredient in moisturizing lotions or creams. And again, next, ceramides have moisturizing property, and fungi like Phalaenophyllae and tuber indicum is reported to produce ceramides. And ceramides can be successfully produced by transformation process using fungal lipase. Using lipase enzyme, we can get ceramides, and these fungi, they are producing the ceramides. And next comes the chitin chitosan. They are copolymer with very effective moisturizing properties, and we know the cell wall has glucan and chitin. Chitin and glucan copolymer, they are very effective moisturizing agents. And this chitosan, from this chitin, chitosan can be obtained. There are also this chitin or chitosan are used in skincare formulation as moisturizing agent. And so far, this chitin and chitosan are obtained from insects, mollusks, and crustaceans. So we know, so this all create conservation problems. When it is obtained in more amount, it will create conservation problems, and it's very difficult to isolate from these things. And we have to first rear it or propagate it, and then only we can isolate chitin, chitosan. But fungi is very easy, and enormously we have this resource in the chitin chitosan available in fungi, and this can be a target for future. And again, we have pigments for in cosmetics, coloring agents. We can have this natural resources, and mostly researched one is monascas, and we had a talk also on this pigments, and safe ones can be researched for usage in cosmetics too. Because this fungi has wide range of pigments. This, some of the pigments produced by filamentous fungi, and safe ones can be used for cosmetics. And not only that, the fatty acids are used as substrates for bioflavor synthesis, and fungi has a role in this too. And now I'll show you some products in the market which is blooming in past decade. And so these are some of the products from mushrooms, mushroom extracts, fungal extracts, which is having demand in the market. So this one, first one, it is from Italia. It is antioxidant and vitamin D product, which is from Ganoderma and Lentinus edodes. And this one, it is from US. It's a cleanser, exfoliating cleanser. Its function is to remove dirt, oil, and makeup and fight signs of aging, they say. And this is from US and UK, from Ganoderma lucidum, Lentinus edodes. And this again from Ganoderma and Inonotus obliquus, Ganoderma, Lentinus edodes, Mucor mehi for anti-aging. From France, products are, and these are from Japan and Spain, and from Cordyceps sinensis, Schizophyllum commune. These products are produced, and these are very few. And now Johnson and Johnson and very leading cosmetic companies are started concentrating on mushroom cosmetics, cosmeceuticals, and we know it is next to insect, the fungal biodiversity. We have the second largest biodiversity, and it is revisited that 3.8 million species is available worldwide. And you might have seen in this previous studies, which all I showed here, the reviews I showed here, all exhibit all common known fungi. See, the products are from Ganoderma, Lentinella idox, and Schizophyllum commune, all already reported and well-known fungi and mushrooms only. The products have come out, but yet we have a huge number of fungi to be cataloged and to be explored for its potential. And surely there is a great future when young researchers select this area of research, fungal cosmeceuticals, when they are doing their MSc projects or for PhD, if they are selecting one cosmeceutical from fungi, it's damn sure they are going to be millionaires with the product when they convert their knowledge to economy. And India too is rich with this, more than 96,000 fungal species, and we have a great future with this cosmeceuticals. And if you select cosmeceutical for your research, um, it will never go vain, because just it has started to bloom, and researchers want it in plenty, because tons and tons of cosmeceuticals are needed, in demand in the future. And if one product is coming out, that will be itself is a great success for the researchers. So thank you for the opportunity, sir, and hope it would have kindled some researchers towards this cosmeceutical, fungal cosmeceutical, and I'm happy to take questions.
Thank you very much, madam. Thank you very much for this wonderful presentation on cosmeceuticals, something which is not very popular. So there was a need to create awareness about this among the mycologists and the people who are interested in applied research, actually, using fungi. Now we'll allow our audience to ask questions. Maybe Ms. Dolma, please ask your question. I'll just unmute people. Ms. Dolma.
Yes, thank you, sir. Ma'am, my question is that people in Himalayas tends to have a fine lines at a very early age, maybe around like 20 or 25 age. So is it effective to use that Avino product like you showed in the slide? Is it effective to use at an early age, or can it be used at a later age also? The people that has already developed wrinkles.
So this actually, now we are getting exposed to environment pollutions and UV radiations. So it does not matter that age is a factor for making this wrinkles and all. So if there is a wrinkle, they can use it. We have some chemicals, and now people prefer organic, that non-chemical forms they are they are preferring now. So that can be that non organic forms can be preferred. And research, if you are taking up research, fungi is having good hope for that too.
Okay, ma'am.
It can be
So
taken.
okay, so this product is not harmful for the younger age, ma'am.
No, it is not harmful, because the products are mostly prepared with edible fungi, already reported safe.
Okay, ma'am. Thank you, ma'am.
Yes, thank you.
Madam Smita.
So, Madam, the presentation was so good. So I have some questions. So essentially, you have highlighted these fungal as cosmeticals. So the question is, apart from the normal individuals, now the challenge remains in treating the patients who are undergoing therapy and different when they are challenged with different diseases. So can these products be used on them, like where their immune system is already compromised or they are undergoing some treatments? So it is isolated from fungal like that. So can these be used on them, or is there any supporting evidence to say that these products are safe even on them also?
Ma'am, actually, if you see the products I showed as examples
Okay.
for fungal cosmeceutical research, so already industries and researchers are on the way on this research to show that. But these products already, this Ganoderma lucidum and all has shown excellent results. There are thousands of reports on its safety, and it is having versatile potential on all aspects. So these are all safe, but very few products are there with this safe mushrooms and fungi. And there are a lot of area for this research. So that was my actual highlight. But what the products I showed also, it's proven actually safe on all immunocompromised patients, or even in all aspects it's proved. This Ganoderma, Lentinus edos, they have lots of reviews and papers on this. But my talk was for opening areas, arenas for researchers, to open a new niche for researchers on these aspects.
Okay, madam. Madam, how long it takes essentially to isolate a novel compound from any of these fungal species and to characterize and then go for testing? Roughly how long the span it takes?
Well, we are also working on some areas on chitin chitos on anti-tyrosinase activity. So it depends. If it is it's a mushroom, because we are working on wild mushroom, so we have to make mycelial cultures and then mass produce it, and then we can go for the extract and test on that. So it will take some time. But if it is directly on, if you are getting plenty of mushrooms in that area, you can directly go for dried products. But anyway, we have to mass cultivate it. So making it mycelial form and then going for the work will be a preferable one. And it takes time, some time. It is not immediate results, or immediately within short span. We cannot say that we will come out with the product, because it is we are working with mycelia. So we dedication on that, really will get successful results with the new cosmeceuticals, because it is just blooming.
Okay, madam, my last question. So the question is, now we see many individuals, they get allergic with on exposure to certain kind of food. So if these products come to market, so will it have impact on certain, individuals who are allergic to certain mushrooms like that? Will it have an impact?
So that
If so, how do you defend it?
only R&Ds will prove that, ma'am. We cannot just haphazardly say that it will have effects or not. Only R&Ds will support your question.
Okay, madam. Thanks for the answers. Okay,
Thank you, ma'am.
thanks.
Mohan Kumar, please.
Madam, I have a question. This is regarding the telomerase activity. Most of your talk is talked about the inhibition of a telomerase enzyme. Some
Tyrosinase.
company,
Tyrosinase
Tyrosine.
enzyme, I said.
Yeah, and there is some of the telomerase enzyme. There are some of the cosmeceuticals. They are adding telomerase enzyme. So this telomerase enzyme, can we obtain from the fungal sources? Are there any, uh, 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 source which can obtain the telomerase enzyme? Because as the age progresses, the telomerase decreases. And
Yes,
there
sir.
are some companies who are adding telomerase into the cosmeceuticals.
Maybe,
Uh,
sir.
yeah, are there
Uh,
any research saying
Till
that?
now, there is no report on that aspect, sir. But certainly, even when you are searching for anti-tyrosinase or anti-elastase, such kind of things from fungi and mushrooms, the works are very, very limited, and very few papers are available. And as you said, there is no reports for telomerase in cosmetics with the help of fungal agents. Uh, but certainly, this may help. Fungi and mushrooms, I can say fungi will have this agents also, which you can take up the research on that area also, sir. Certainly, it will also have benefit.
But any specific compound is giving an tyrosinase, or is a consortium of the compound, ma'am?
So
Anti-tyrosinase.
reports are there for individual compounds also, sir. Extracts are giving effect, individual compounds, polysaccharides. So the reports are there in all aspects. So individual compounds are also having effect.
Okay, thank you, ma'am. Thank you.
Yes, sir.
Dr. Praveen Kumar, please. Ms. Dharma, please mute yourself. Otherwise, I will remove you from the meeting. Thank you. Dr. Praveen Kumar, please. Siddharth Anand, please.
Yes, hello, am I audible?
Yes,
This
you
is
are.
Siddharth. Yeah, ma'am, I had a couple of questions. One is that when we do phytochemical analysis of all of these, what we call medicinal or cosmeceutically important mushrooms, we see that there is activity in the aqueous extracts, the organic extracts. So is it possible to go ahead and try and make a product with holistic extracts, or do we have to always isolate specific compounds and then go and make a cosmeceutical with it?
So it depends. Now demand is there for holistic also. Here all mushrooms which I have shown is already reported as edible mushrooms, safe mushrooms. It's already reports are there and they have worked on that.
Right.
So if you are taking new one which is not reported as edible, or its compounds are not reported yet, you may go for in silico studies and some versatile approaches you can plan accordingly. But now even for patents you can go as holistic also.
Okay.
Uh, it is preferred. Or if you think that single compound is going to have so this your mushroom or a fungi is having a single compound which is going to give better results, or it is having a very high potential, you can go for that, or you can prefer holistic approach and proceed with.
Okay, ma'am. Ma'am, my next question is that you mentioned that you are leaning towards mycelium cultures for production of these compounds that of interest in your research. But most of these are secondary metabolites, right? So they are generally produced only after the fruiting body is formed. So is there a way in which you go about it to get these metabolites in mycelium or the vegetative state?
Yes, sir, we tried with Odamenciella and Pleurotus also, sir. Both mycelia is having very good anti-tyrosinase effect. And this
Okay.
collagen, yes, sir, chitin, chitosan. So this cell wall has, fungal cell wall has those
Right,
things.
right.
So
Okay.
it's not that only fruiting body can have these effects. So mycelia also we can go with,
So
and it
it
had
depends
good
on the compound.
yes, sir.
Okay, ma'am. And ma'am, just one last question. Nowadays, microprospecting has become like the new buzzword, right? Where everyone is looking for newer and newer mushrooms that we can isolate novel compounds from. So, is there a way to go about it, like whether there's a pharmacogenetic approach or an oncogenetic approach that people are adopting, like after like getting some, let's say, no,
वो
uncatalogue the wild
that
mushrooms
is very
from,
disheartening. मैं मतलब
sorry?
Sir,
नहीं
I could
किसी ट्रेनीज वगैरह से ज्यादा वो रखना भी नहीं है.
Madam boss, please mute yourself. I will remove. Otherwise, I will remove
नहीं
from the
एक्चुअली
board.
वो यूज कर लेते हैं ना अपना टाइम पे, इसीलिए
Oh,
मैं
yeah.
अंकिता के लिए भी थोड़ा स्केप्टिकल हूँ. वो भी उसी फैमिली को बिलोंग करती है ना, कैसी होगी गॉड नोस. ऐसे नहीं देखना चाहिए किसी को. एक
Sir, I couldn't hear that question because I had a crosstalk.
Yeah,
Yes,
please go
sorry.
ahead.
Yeah, I'll just repeat my question, ma'am.
Yes,
So,
sir. Yes, sir.
I was saying that is there a way of looking for these specific type of compounds? Like, is there an oncogenetic approach, or whether there is a phylogenetic approach? Like, certain families have certain compounds. Certain genera are better to look for certain types of like collagen-based compounds, or is this just purely experience-based, where we get each specific mushroom or mycelium and then we do a complete analysis from it?
So that also can be done, but actually this anti-tyrosinase effect, tyrosin is present in almost all organisms. So which one
Okay.
is best? Which one yields best? Which shows better effect? That we can screen. We can go for. So like for example, we'll have cellulase producing organism, protease producing organisms. Industrial level production, we'll go for screening, primary screening, secondary screening. Same thing we can go here because chitin chitosan is there and anti-tyrosinase effect is there.
Okay.
So we can find which one is better by screening.
Okay. Thank you, ma'am.
Yes, sir.
Ms. Nagendra, please. Pratinay, please.
Hello, ma'am. Thank you for this informative session. I wanted to know whether the anti-aging cosmetics, whatever natural products we have, will the potency differ with each person, or maybe because of the region or something like that? Will it differ depending on the region in which the person is staying?
Certainly, ma'am. There are intrinsic factors, extrinsic factors. So, intrinsic factors, it has even genetic reasons for the production of enzymes, or for the factors affecting uh, aging or whatever it may be. There are factors genetically, and the environment also plays a role. So, when we are in the environment polluted, so our the response is different. So, certainly there is difference from individual to individual.
Okay, ma'am. Thank you.
Yes, ma'am.
Dr. Sanjay Saxena, please. Malika Sudhar, please.
Good afternoon, ma'am.
Thank
Thank you for such a wonderful talk. And I would like to ask, ma'am, that melanin is also a pigment synthesized by fungi, and fungal melanin is also widely used in cosmeceuticals as sunscreens. So, can you please give some insight regarding the relevance, or how much is fungal melanin incorporated in sunscreens is a success?
So actually, that was my first area of the talk. Uh, Actually, anti-tyrosinase effect is the target. Actually, the tyrosinase enzyme will actually promote melanogenesis. So melanin production is through tyrosin. So anti-tyrosinase effect. So when the creams and lotions are incorporated with anti-tyrosinase enzyme, so it will give effect. It will limit the pigment production. So that is the target, and there are so many mushrooms produce that. They have come out with creams and lotion with those extracts, and some single compounds also.
Okay.
It's very effective, and we can proceed with further some known fungi also.
Okay, okay. Thank you, ma'am.
Yes, ma'am.
Dr. Sanjay Saxena, you want to ask your question?
Yeah, I'll take this question. Madam, very informative talk. I just was wondering that if we could explore phytotoxins and mycotoxins on the same line as botulinum toxin has been used for skin tightening as a cosmeceutical application.
Sir, I'm not audible. It's very feeble. The voice is very feeble. Can you?
I'll speak again, madam. The talk was very informative. I
Yes, sir.
wanted to know that if it is possible to use mycotoxins and phytotoxins on the same lines such as the botulinum toxin being used for skin tightening.
For
Skin tightening. Botulinum toxin is used for skin tightening.
skin tightening.
Yeah.
Yes, sir. But for skin tightening, toxin studies I didn't come across actually. So I had thoroughly checked, because I made a book chapter for agrobias book. So I went through the review elaborately, but I have not come across with toxins for skin tightening.
Okay. Thank you very much, sir.
Please.
Ma'am,
Nice,
this is Dr.
ma'am.
Shivaranjani. Ma'am, very informative session.
Madam Shivaranjani, please wait. I have to allow other people now. Thank you. Dr. Ikechi, please. Dr. Madam Mantarani Mahapatra.
Thank you, ma'am, for the informative session. Actually, I am very interested to come for this skin pigmentation, and I want a queries have that if you are targeting inhibition of elastase, collagenase, and hyaluronidase, so any side effect in short term or in long term? Is there any
That we have to check, because it will be in different levels. So when we take it to final product. So before that, you can go for laboratory studies, finish your studies in laboratory level. Then we can patent it, have license with some pharmaceutical agents, and you can further go for elaborated R&D studies, and then come out with the product. So, but it is reported to have it is safe to have anti-elastase, this collagenase incorporation in ointment and lotions, anti-aging.
And can, ma'am, any compound that enhance the synthesis, because that is also an alternative if we are enhancing the production of that elastin or collagen?
No, actually, we have to suppress them, not make them increase, because these enzymes help in degradation. When it is degrading elastase and collagenase, it will lose the structure. So the structural support is given by these compounds, and when it is degraded, it will lose the structure. So we have to have anti-elastinase and anti-collagenase enzymes. So some mushrooms, they have already reports are there which is having this anti-elastase and anti-collagenase compounds in it, and that is now explored for further.
Ma'am, I think if it is 50% degraded, like from 100%, then how can we recover that 50% degradation of the collagen or elastin?
No, actually, it is if it is 50% degraded, we have wrinkles and this, and further prevention can be done with this, because it will prevent the degradation or decrease of collagen or elastase. So that is its role. So further, it will protect.
Okay, ma'am, and which cell line we should use for screening? Like which cell line? Keratinocytes.
So normally they go for the normal cell lines and dermal cell lines.
Okay.
Okay. Thank you so much, Madam Mamta, for your questions. Last question from Dr. Ikechi. If can you hear us?
Yes, I can. Good morning.
Yeah, good
Thank
morning.
you for the informative teaching. In as much as we can find use mushrooms to produce these cosmetics, have you also come across some filamentous fungi growing on some of the cosmetics, especially facial powders?
Ma'am, can you repeat?
I said, have you found some of the filamentous fungi growing on facial powder? Have you
facial powder?
Come across it? Have you done any work on it?
So filamentous fungus reports.
Have you found fungi growing on them? Filamentous fungi.
Yes, ma'am. Yes, ma'am. So in my presentation also I showed the filamentous fungi, the reports on filamentous fungi for antiterrorisnase and for fatty acid production, Rhizophus aspergillus. So they have reports and their products are incorporated in cosmetics.
All right, thank you.
Yes, ma'am.
Thank you, Madam, for the question and the answers. The last question from Professor Bhatt, then we will break for the lunch.
Yeah, and I just wanted to say it's an excellent talk, Professor Usha.
Thank you, sir.
I'm very happy to hear this talk. And you are working in a truly a nice place in Kodai Hills.
Yes, sir.
I did some work long ago, mainly on microfungi in 1970s and 80s, some collections, and we did a great deal of work when I was in Madras, along with the Professor C. Subramanian. So my appreciations for your work and your presentation. Great job you did. Thanks very much.
Thank you, sir. We will invite you to our station, sir, once, because it is truly a treasure. The area is a treasure, and we'll have a lot more to explore here, sir.
Yeah, Kodai Hills is a wonderful treasure. Thank you. Thank you very much.
Thank you, sir.
Thank you.
Thanks a lot.
Thank you, sir. Thank you, ladies and gentlemen. I must stop this discussion. I mean, this discussion can continue later, maybe on WhatsApp groups. Madam is there on one of our WhatsApp groups, maybe MycoAsia International. Okay, thank you. You can catch her there. Madam, thank you very much for spending your Sunday with us and for giving an informative talk on cosmeceuticals. Thank you for patiently listening to all the questions, answering them. It has been a wonderful experience spending time with you, listening to you. Thank you very
Thank
much.
you, sir. Thank you for the opportunity, sir.
Thank you. Rohit Sharma will give the final comments before we break for the lunch. Rohit.
Yeah, thank you, Dr. Krishna. Thank you, Professor Usha, for a very nice and informative talk. Definitely, there is a need to work towards the applied aspect of the fungi. And apart from the cosmeceuticals, nutri-cosmetics are also coming into things, and people are getting interested. And I was just seeing the data that it says that 1.8 billion US dollars business is the fungal cosmetics itself. So thank you for very nice details that you have provided regarding the anti-tyrosine activity and the anti-aging potentials of and other chitin and chitosans produced by Ganoderma and lentinas. One of my collaborator from Malaysia, he has also established a company called Kera Grow, and he has got a funding of around 8 crores, which is producing these keratin proteins from and anti-aging compounds from the feathers using the fungal enzyme. So really, it's a great field to work towards the applied aspects. Thank you, Professor Usha. Thank you for the time and the information that provided through the talk. Thank you very much. I'll not be in middle of the people's lunch much. So I think, Dr. Sharma, we can break for the lunch.
Yes, we'll break for lunch. We'll come back in 45 minutes at 2 p.m. Indian Standard Time. And I request the audience, some of you probably experienced that some people are creating ruckus.